And the next speaker
I have to wire myself up.
Stefan Treuer from Göttingen, and we're shifting gears. Stefan is the neurophysiologist among the speakers today, and he's going to talk about single units, I guess, motion processing and attention. So Stefan, thank you for coming.
Thank you. So yes, my presentation will focus on electrophysiology from awake rhesus monkeys, but I hope I will be able to show that there are quite a few links to the previous talk. I will talk about how information processing is modulated. I won't do it in the time axis, but I think you will find many parallels in terms of the spatial and other dimension axis where selection plays a central role in the visual system, and which I think, at least I found the last talk very stimulating in that sense of the connection. Okay, so I will focus on visual information processing along the dorsal pathway in non-human primates. And I want to state something obvious at the beginning, that when we talk about sensory information processing and sensory information input and motor output, we do need a joining element in between. And the joining element in between is the internal representation that we generate based on the physical sensory evidence from our environment. And ultimately, we base our perception and action on that internal representation. That, of course, is nothing new. Artists have known this for a long time. So here you see an artist rendition of a representation. René Magritte drew the overall painting, and of course he shows here what a representation could be like. And I'll come back to this at the very end to question whether this is an appropriate model for visual perception or internal representations in vision. Now my interest, as Udi was pointing out, is motion processing, which is a very important ability for our everyday survival. The ability to very quickly, efficiently, and reliably process motion information, I think, is beautifully demonstrated in this video here. You can see that the error rate of an animal like this over a lifetime should be extremely low. You know, you're 10 meters above the ground, the trees are constantly moving, you're constantly moving, and you really have to grab the right target every time. You don't have much tolerance here. And that, of course, creates an enormous evolutionary pressure on sensory and motor systems to be able to deal with this effectively. And part of the effect of evolution has been that primates, non-human and human primates, have developed a very sophisticated visual system. And we heard some of the aspects today. Also for the auditory system, that is true. Now, if we have a highly developed visual system in this case, that's wonderful, that's great. But think back of what you just heard in the previous talk about what happens if you get too much information. You do have a problem because you have to start being selective about what you can process from that mass of information that's coming in. And I just want to illustrate this with a picture like this. Now, from your retina's point of view, that picture is not a challenge. There are 120 million photoreceptors. They pick up that information immediately, and it's right there. And if I take it off again, you know, the cells are ready for the next image. Of course, for us, processing all of the information that's present in that image in one instance of time is essentially impossible. We need to be selective. So these powerful sensors that we've developed over evolution, they're great, but they really create a need for selective processing. And that selective processing is usually called attention. And fortunately, everyone knows what attention is. So I could stop here, but I guess I have a couple of things I want to say. But even in 1890, to William James, it was clear two things. One is that attention is about selection. That's the thing he talks about first. But I think even more importantly for everyday neuroscience is the last sentence, that attention implies withdrawal from some things in order to deal effectively with others. So we get a benefit of being selective, but it also means that we lose our ability to process the non-selected aspects from our sensory information input. So for the context of my presentation, I define attention as the selective modulation of sensory signals based on their behavioral relevance. So we pick out certain aspects at a given moment based on why we're processing that information, and we treat some of these aspects different than others. That's the behavioral relevance influence on the selective modulation. If we want to do this in a model system, one of the model systems that I've been working on is area MT in the visual cortex of rhesus monkeys. So why area MT? Now, area MT is one area in the processing hierarchy, and that we're here to talk about processing hierarchies. One of the processing hierarchies called the dorsal pathway or the parietal pathway, which takes information from primary visual cortex and feeds it through to higher areas. And area MT is particularly specialized for motion processing. It's chock full of neurons that care about the direction and speed of motion in the visual environment. And so it seems like an opportunistic place to look for an interaction between efficient and precise information processing from a sensory point of view and possibly influencing that processing based on attentional selection or attentional modulation. So let's for a moment stick to the sensory aspects of processing in area MT. Now, as I said, area MT is characterized by direction-selective neurons. They are usually considered the neural basis of motion perception based on lesion and stimulation studies. And the characteristic elements of these neurons is that they have well-defined receptive fields. So this is sort of symbolic for a typical MT receptive field. If this would be your fixation point, your line of gaze, then you would have a receptive field here, and of course covering the whole visual field like this. To stimulate these neurons physiologically, essentially what you need are moving stimuli. It almost doesn't matter what you use to stimulate them as long as the stimuli are moving. But they're direction selective because the direction of movement matters. So a typical response of an MT neuron would be plotted like this. This is direction of motion. This is strength of response from the neuron. You get usually these nice bell-shaped tuning curves. This one here would indicate the preferred direction of the neuron. So in this example, rightward motion. And then you have a characteristic drop-off of responses. And in this case, leftward motion would be the null or the anti-preferred direction. This is the typical sensory response, treating MT as a bunch of filters that take a particular input, being direction of motion, and generate a well-defined output. And of course, that has been extensively investigated to measure neural responses, compare it to visual responses and perceptual quality, and there's a very high match between area MT and overall perception. Now, the reason I'm here today is I want to talk about an additional aspect of these neurons, and that is going beyond the sensory representation into the question of how attention modulates this relationship between the stimulus and the response. And I'm going to do this for the next few slides based on single stimuli representations in area MT. And some of this is work that's been done quite a while ago, but I think it's an important basis for some of the newer things I want to point out. So let's think about a careful experimental paradigm. What we want is a situation where we have high control over the stimulus, and then we want to modulate responses that are not because the stimulus is modulated, but because the attentional condition is modulated as a requisite for demonstrating attentional modulation. So the paradigm we typically use in my lab is to train rhesus monkeys to fixate a fixation point on a computer screen, and in this example here, to present two moving stimuli. And the task of the animal is to respond to changes in an otherwise continuous flow of these random dot patterns. You see every now and then there's a small change in the direction of motion. Now the animals can be cued in advance to either attend to this stimulus or to this stimulus, but always both stimuli are present. That way we can generate two situations in which the physical input of the system is identical, but the behavioral state, meaning where and how attention is allocated, varies between the two conditions. And if we see a modulation in responses, it's based on the attentional condition, not on changes in the sensory input. So let's look at what happens if you take this scenario here and you place one of the stimuli in the receptive field of an MT neuron. So here's a symbolic representation of this. Again, fixation point. This would be the receptive field of a given neuron. These would be the two stimuli presented. And you can now recover the tuning curve of these neurons by showing different directions of motion inside of the receptive field. So in this particular experiment, the two stimuli always move in the same direction. But of course, between trials, you can change that direction. So essentially, 8 to 12 directions are used. And we can then recover the tuning curve. So this tuning curve is it's just an example neuron, direction of motion here, responses here. Now I painted in green because this tuning curve is measured when the animal is attending to this stimulus over here. The response, of course, is still the response to the stimulus inside the receptive field because that's all that the cell essentially sees. But we would call this the unattended tuning curve, because the stimulus that the cell is responding to is not attended. This is the attended stimulus in this condition. Now we can change the instruction to the animal, ask the animal to attend to this stimulus without changing anything else, and remeasure the tuning curve. In this example here, you see sort of a prototypical effect of what we have observed in area MT. An overall increase in responses. Tuning width is essentially unchanged. It's like a multiplicative scaling of the responses. It's like a gain change to the neuron. And most importantly, it doesn't narrow the tuning curve, as has been hypothesized based on some other data. It just scales them up. We will call this a gain modulation. On average, across our population of neurons in this particular study, we had a modulation of about 10%. It is highly significant, but quantitatively it seems like a relatively small effect, but I'll come back to whether 10% is a large or a small effect a little later. Now, what that shows is that you can base attentional modulation on a spatial selection, because what the animal is doing here is selecting a stimulus based on its location. Okay, and that's what's different between the two conditions. So that's a spatial attentional effect. What we were wondering, though, is whether there is also a possibility to modulate information processing not based on spatial location, but based on the feature. And with feature, I mean something like direction of motion, color, or shape, or something like this. So for MT, we use direction of motion. So here are two conditions to compare. In the receptive field, stimulus moving upwards in this case. In the receptive field, stimulus is moving upwards. Outside, the stimulus is either moving downwards or upwards. In this example, the neuron actually prefers upward motion. And of course here, because it's the same neuron, again, it prefers upward motion. In both of these cases, attention, spatial attention, is outside of the receptive field. So we're measuring the response of the neuron to a preferred stimulus in both cases, unattended in both cases, and we were wondering if attending to the preferred or null direction outside of the receptive field would modulate responses. Why would we wonder whether this happens? If you think how we use feature-based attention in everyday tasks is we use it to select or pre-select information based on a non-spatial information we have. So for example, if I want to find somebody in a crowd like here, and all I remember is that he's wearing some orange type t-shirt, I cannot use spatial attention because I don't know where the person is. But if I would have a selector that enhances all orange representations in my environment, the person would pop out, it would be more visible than other people, and I would see him in the back there. So that would work. Physiologically, this would mean, though, attending to a particular direction needs to have a global effect on the neurons, not just locally where I'm attending, because I want to spread that type of attention. And in fact, that's what we find. We find that neurons get more responsive in this situation than in this situation. So this neuron becomes more driven or more active if the attended direction matches its preferred direction. So if there is a match between what I care about right now and what the neuron is good at encoding, that's the condition where we get a higher firing rate than when I'm attending to something that the neuron actually doesn't particularly care about. And the effect is about the same size as what we find for spatial attention. Now, as I said, these numbers are not particularly large. It still means the neurons are responding even in an unattended condition. But maybe we can combine these two types of attention to see whether they're additive or whether there is some sort of limit in an area like MT to attentional modulation. So here we combine them. Attention outside versus inside. So spatial attention from a low response to a high response. Feature-based attention from attending to the null direction to attending to the preferred direction. So we're trying to pick the two extremes of the combination. And in fact, the attentional modulation adds up. So you get a bigger attentional modulation if you combine the two types that I just introduced you to. And it seems to me an almost a linear combination of the two. So attention seems to reflect the sum of all selections that work on a particular situation. If many types of attention are combined, you get particularly strong modulations. And we try to capture this in something I hesitate to call a model. It's more of a descriptive way of dealing with our data. And the idea behind the feature similarity gain model is to predict the attentional modulation you might observe in a given situation. And it's really only based on two factors. One factor is that it's a gain change. I showed you that at the beginning with the scaling up and down of the tuning curve. But if you want to know whether this gain change is large or small, or whether it's a suppressive or an excitatory effect, all you need to look at is the feature similarity. Now, what do I mean by feature similarity? I mean it's the similarity between the attended feature, now that could be location or direction or orientation or whatever you want, and the feature preference of the neuron. Every neuron has a spatial preference, that's its receptive field. So that can match or might not match your spatial attentional location. And these neurons have a preference in terms of direction of motion, and that might match or might not match your current behavioral target. If both of them match, you have great feature similarity. If neither of them matches, it's very poor, and you can have any in-between combinations. So that is a nice and pretty complete description of what we observe physiologically. And in a way, for the more computational types amongst you, gain changes are nice because gain changes are easy to handle. But then you also know that gain changes are limited because you can't do all that much with gain changes. All you can do is crank up and down responses. And particularly, they seem to always generate multiplicative effects. Now, there are examples of non-multiplicative attentional modulations, and of course that creates the question, how can this be? How can there be a gain change system and we can still see non-multiplicative attentional modulations? And that's, I think, an important and interesting question that goes beyond attention, and it's the relationship between what we record from single neurons and the situation that we actually deal with in everyday life, which is not that we only have access to a single neuron in a given situation. We have a whole population of neurons. Many of them will respond in some way to the stimulus. So does that change anything? Well, in the usual approach, it doesn't. So usually, if we look at a population response based on single neurons tuning curves, the two are actually a match, at least if you assume that you're recording from a typical neuron. So why do I say that? This is the curve I showed you before. Stimulus direction versus response. This is a single neuron tuning curve. Now, if we assume that this is a typical neuron, what would be the population's response to a single stimulus, but many neurons tuned to different directions? Well, actually, it would look exactly the same way. The only difference would be the axis is not stimulus direction anymore because you're only showing one stimulus direction. The axis would now be population of neurons preferring leftward motion and their activity, population of neurons preferring up and to the left, population of neurons liking rightward motion, and so on. And you would only switch the label on the axis. So in one case, it's stimulus direction for a rightward preferring neuron here, and in the other case, it's the preferred direction of neurons given a rightward stimulus. So it seems that anything we can show with single neuron responses, we can immediately say this is the population response. And therefore, we can say that attention has a multiplicative effect on population responses because I showed you that it has a multiplicative effect on the single neuron. But this is where the analogy breaks between these two systems. And this is because attention cares about the relationship between the stimulus and the neuron, or the attended situation and the neuron. Remember, that's feature similarity. So if you apply feature similarity to what I just showed you, let's think this through. So there's a rightward stimulus here in the environment, okay? And the person or the animal is attending to the rightward stimulus. Rightward preferring neurons, for these, this will be good feature similarity, right? You're attending to what the neurons care about. Now, the feature similarity gain model would say, let's go up here because I'm increasing the gain. But what about these neurons here? Same situation. These neurons prefer leftward motion. You are attending to rightward motion, which means poor feature similarity, predicting a suppression of responses. So even though every time attentional modulation is just doing a gain change, the gain change would be different for this group of neurons than for this group of neurons than for this group of neurons, okay? So if that is true, what I'm saying, even though the system only changes gains, the result is a population response that is non-multiplicatively modulated. So we actually looked at this situation And without going too much into the details, this is what we see here. The curve I just showed you would be sort of a neutral condition when the animal is just fixating, and this one is the one where the animal is attending. And you see exactly what I just explained. An enhancement in the center, a suppression on the sides. This is clearly not a multiplicative change. This is a non-multiplicative change, even though in the assumption is that everything that happened here was always gain changing, but gain changing depending on the feature similarity. And that, of course, opens up a lot of possibilities in the system or in a network with simple gain changes. Now, if attention enhances the representation of attended stimuli, you might want to think of this like an enhanced contrast, which, and I'll come back to this later, as a sort of a very general feature of any visual stimulus. It has some luminance strength in the visual system. And maybe attention actually enhances that perception or that representation. But I want to point out that there might be other changes, other perceptual changes that happen with attention. Let me walk you through that question. So I showed you this sort of typical example of our experiment, right? So this is what the animals are seeing. They're seeing these changes. Now, if attention would simply enhance, the visibility of the stimulus, this wouldn't necessarily help you with the task, because your task is not to detect the stimulus. Your task is to detect that change in direction. Now, easy to recognize changes would mean big changes. Hard to recognize changes are small changes. So ideally, if we are not thinking as a physiologist, ideally, you might want to hope that attention makes the change look bigger than it is, to make it more detectable. But physiologically, that's actually not that trivial to make, some parameter of a stimulus just become larger without physically changing the stimulus. But we were wondering if this was happening in our neurons or not. So how do we look at this? Let's switch to a view of these neurophysiological data that I haven't shown you before. Here's a sort of a extrapolated population response over time. So this is the course of a trial. At point 0, the direction of motion is changing, and at 200 something, the direction is changing back to the starting direction. And this is sort of an assumed population of neurons based on our data, aligned to their preferred direction. So what this would mean here is that the group of neurons that encode the stimulus that's present are aligned up here, and the others are here. So essentially what you're seeing here is that population tuning curve that I showed you before. Okay? Now, not much surprising is happening here. You show a constant stimulus. What you see is a constant profile with a high activity level close to the preferred direction of the neurons and low activity out here. Stimulus direction is changing, population response is changing, stimulus is changing back, population response returns. Okay? So that's exactly what you would expect from a population of neurons encoding the direction of motion in the environment. Now, what we can do now is look at the exact same condition for the attended stimulus. This is for the unattended stimulus. And what you see here is an enhanced response. That's what we expect, given that we know the stimulus is more powerful. Still see the change in direction. Everything looks fine. But let's zoom in a little bit to get more quantitative. So if we zoom into that region of change, this is what we see. So I now drew in white the physical direction of the stimulus, starting out at 0 degree, jumping to 25 degrees, staying there, jumping back. And you sort of see here the smooth population response for attended and unattended. And the black curve is a fit through the peak of that population. Sort of, what is the population telling us is the presented direction. It matches very well when the stimulus is constant, but when the stimulus changes, there is an overestimation. So the population slightly overestimates the change in direction. And for the attended condition, that overestimation is even larger. So what we find in our data is, yes, basically area MT is encoding the physical direction in the environment, but at least in a task where changes in directions matter, whatever happens here is it changes or enhances the change that's behaviorally relevant. And that's quite a substantial effect here. It's about 13 degrees overestimation. Usually we have direction discrimination threshold in the 1 to 3 degree size. So that's a substantial overestimation. We're currently running psychophysical experiments to see whether perceptually the same thing happens, whether perceptually we also overestimate the direction. But the monkeys don't have to respond to the exact perceived direction, they just have to say they saw something changing. So that suggests that attention not only simply makes a stimulus stronger, but adapts that strengthening to the behavioral condition that are currently ongoing. Now, we are talking about hierarchies today, and everything I showed you so far is about area MT. That's not very much of a hierarchy. That's just one level of the system. Now, one very obvious situation in attentional research is that the effect of attention grows as we move through the hierarchy. So when I started in this field, not even MT was assumed to have any attentional modulation. Nowadays, even primary visual cortex has attentional modulation, but it's substantially weaker than what you usually see in extrastriate cortex. What we wanted to look at is whether the main effect of moving up the hierarchy is somehow related to attention. And the main effect is that the receptive fields grow. They grow along this pathway and they grow along this pathway. So that means we're moving from a system here that is really focusing on tiny details in our spatial environment to areas that are covering almost all of the visual field. So neurons that have to integrate a lot of information and neurons that look at the fine details. And we're at an intermediate stage here and here where receptive fields are substantially large. And the one thing that happens if receptive fields grow is that they often cover more than one stimulus. In V1, you can pretty much assume no matter what you do, there's one stimulus in the receptive field. In these higher areas, you now can ask whether things happen inside of the receptive field. And one of the questions we can ask now is what happens if you have an attended and an unattended stimulus in the same receptive field. Now, spatially speaking, now the receptive field covers both. And just turning on or off a neuron will not help you in that situation. So let me sketch out what the stimulus look like. So for example, we can use, instead of the circles that you've seen before, we use two half circles moving in opposite direction, and the animal can be asked to attend to one or the other. And we can see whether this changes anything, because spatial attention stays within the receptive field. These random dot patterns that you've seen before have a nice feature. We can make them overlap totally. We can generate two stimuli at exactly the same spatial location by just moving half of the dots in one direction and half of the dots in the other direction. That would be this stimulus here. So you see the only difference is here spatially segregated and non-spatially segregated, both of them inside the receptive field. And we can ask whether attending to one direction in this condition versus attending to the same direction over here makes any difference. Now, we can't do tuning curves here anymore, but what we can do is align one of the directions to the preferred direction, the other one to the anti-preferred, and the same over here. So practically speaking, we're comparing these situations here. Red is the attended stimulus. Of course, they're all black and white on the screen. Ask the animal attend to downward motion or an anti-preferred direction, attend to preferred, anti-preferred, and preferred in a separated and in a transparent condition. Now, in the case on the left here, we get a very large attentional modulation. This is substantially higher than what I've shown you before. Of course, in every population of neurons and every animal, it slightly varies, but this is roughly 4 times the size of what you've seen before. And now let's look at the one over on the right side. It's pretty exactly half. So there's almost no difference here, and yet the attentional modulation is half. But if you think of it from the perspective of what I talked to you about spatial and feature-based attention, maybe that's not so surprising anymore. If I describe what the animal is doing or can do in this situation, it is entirely based on direction of motion, right? Over here, it's based on direction of motion and spatial location. I have 2 ways of selecting the stimulus over here, meaning I can pick the left or the right, and I can pick up or down, and here I only have half. So what seems to explain this difference is that here I only have feature-based attention. I can only use the feature that I'm attending to. Here I can use the feature and spatial attention. And just like what I showed you for the single stimulus case, it seems that these two can be linearly combined. They're approximately of the same size, and they are very well behaved. But if this is the case, if attention inside the receptive field can differentiate between 2 stimuli, this is not about turning on or off a neuron when spatial attention is inside. Something else must be going on here. And hold that thought for a second, because I'll come back to it. That's the one aspect. The other one is I wanted to come back to this issue of contrast or stimulus strength. If we believe that attention somehow affects our perception, and if we see attentional modulation like this, then we would expect that there is some sort of effect on our perception of an attentional enhancement of a stimulus. Now, what that means is maybe I can generate a situation, a non-attentional situation, where I get these kind of response changes in neurons, not because of attention, but because of changing the stimulus sensory-wise. Why is this relevant? Because it gives us an idea of how to compare a 10, a 20, a 30% response change in a neuron to some behavioral effect. Because if I would tell you a stimulus has just become 20% more effective for you in terms of firing rate, would you be impressed or would you be non-impressed? It's very hard to know, right? What does this mean, 20%, 30%, 40%? If I would have said to you, the stimulus has become 20% less visible, now that would be something that we can relate to and that we can call large or small. So what I'm going to try to do is I'm going to try to mimic the effect of spatial attention by changing the stimuli. So no change in attention, just changes in stimuli. So conceptually, this is the idea. Let's take this as our baseline. The animal is attending to the fixation point. There's enhancing and a suppressing stimulus inside of the receptive field. You get some baseline firing rate. What we know is that if you direct attention to the preferred stimulus of the neuron, the responses are going to go up. If you direct the attention to the null stimulus, responses are going to go down. Okay, we already know that. And we know that that effect in MT is roughly 30% plus and roughly 30% minus. Now my question is, can I generate a sensory condition, so with the animal fixating, where I change the luminance of one stimulus to also enhance responses by 30%? Or reversely, can I lower the luminance of the excitatory stimulus to also suppress responses by 30%? If I can do that, then I can sort of say this is what attention does. It enhances or suppresses stimuli in approximately this order of magnitude. Okay? So to plot the data, I want to compare attentional modulation of firing rates, and I said they're roughly around 30% here, versus a decrease of luminance of these stimuli. If they're one-to-one thing, then the data should all lie along a diagonal. Instead, all the data are squished to the right side of the graph. That means even neurons who show, let's say, a below average attentional modulation of 20% in firing rate require a massive change in the physical luminance of the stimulus to achieve the same modulation. You can either call this interesting or not surprising. For those who call it not surprising, you say that because MT neurons are highly sensitive to contrast, they quickly saturate. So of course it will require a large change in luminance before the responses drop. But I would still argue that this is the condition under which attention works. And this is the condition where attention lowers firing rates or enhances firing rates by 30%. And that's why these numbers are, from my perspective, quite surprising. So that gives us an idea that you can't directly convert firing rate changes into attentional effects, and it also gives us a feeling that maybe perceptually the strength of the stimulus is changed by attention. But it leaves us with the other question I was asking you to hold in your mind, and that is, if there are two stimuli inside a receptive field, how do you get a neuron to only respond to one and not so much to the other? I'm going back one slide. So how do you get a neuron to treat two physically equally strong stimuli as if they are of different strengths? Because that's ultimately what the neuron is doing with attention. How do you get this to happen? And there is for a long time, there is a theory, has been a theory about this, proposed by Desimoen already in the last century, about changes in the shape of receptive fields. Everything I showed you so far has this constant receptive field. But maybe attention can change the shape of receptive field and make the cell more spatially selective. And here's a quote, 1985, quite a while ago. And what they claimed, or what they suggested based on data that they've collected from area V4, is when attention is directed to one of two stimuli in the receptive field of a V4 cell, the effect of the unattended stimulus is attenuated almost as if the receptive field has contracted around the attended stimulus. So the data set they've collected is very similar to our MT data set, even though this is the other pathway. And they made this hypothesis, but never actually followed up on it and tested whether changes in the receptive fields actually occur. So that's what we did. We decided to map the receptive field of these MT neurons under spatial attention condition. What does that mean? It means, if you consider this the fixation point, you have various positions where you briefly flash a stimulus and you take the response that you get as a profile of the receptive field. A heat map might look like this, ideally speaking. So this would be the sort of center of the receptive field. This would be the overall receptive field. And you get a map. Okay, this is the neutral map with the animal just fixating. And the question is, if now the animal is attending to one or the other location in this receptive field, is that receptive field, the spatial selectivity of the neuron, now somehow focused on that location? So here we can just compute a receptive field, and we can do the same thing when the animal is doing a task. So here's a sketch of the task. The details are not so important. The important thing is the animal is either attending to one or the other location inside of the receptive field. And while the animal is waiting for the change in the stimulus, we can map the receptive field. So here's an example neuron from the study. This would be the condition when the animal is simply fixating. And these are symbolic representations of the two locations for those two stimuli. And you see both of the stimuli inside of the receptive field, and this is sort of the mathematical definition of the receptive field in this condition. Now, let's keep everything the same except the location of spatial attention of the animal and remap the receptive field. And even though I'm not telling you where the animal is attending, I would hope that you can see it in the data. So there's a clear sign that the receptive field changes when the animal is attending to this region here. The receptive field has sort of shifted over there. And if the animal is asked to attend over here, the receptive field shifts toward that location. Okay? So receptive fields do change, spatial receptive fields change with the location of spatial attention. If you subtract these two maps from each other, then you see what sort of happens. And that is, attended location becomes more effective, unattended location becomes less effective. It's like a push-pull effect on the overall profile of the receptive field.
But this one on the lower left looks very much like the neutral one, doesn't it?
The lower left looks more like the neutral one than this one. Yeah, so that's because it's physiological data. So you get, of course, a variety of effects. In this particular one, it shifts quite a bit over here, and it seems to shift less here. But if you look at the actual center, which is something here, and this one is pretty much in the middle, here it's somewhere there, you see the overall effect. But that's why we have to look across the whole population. This is just an example neuron. I can show you the population data. So how do we quantify this? Let's call this distance here, that's actually the attentional shift, right? The animal shifts attention from here to here. The receptive field responds by shifting its center, well, roughly about here to about here. So that's a different number. And what we want to know is whether the sign and the magnitude of the shifts of these two, behavioral and physiological match. So here's a symbolic representation. There's a stimulus distance, which is the attentional shift, and there's a center of the receptive field shift, which I call the neuronal receptive field shift. If we look at this for every neuron, and that follows up exactly on your question, then we find that almost every neuron has a positive value, meaning that it shifts in the same direction as the behavior of the animal, right? So if the animal shifts from right to left, the receptive field center also shifts from right to left. That's the mean, and the mean is about 30%, which means if there's a 3-degree distance between the two stimuli, the receptive field center shifts by 1 degree. If it's a 6-degree difference, then the receptive field shifts by 2 degrees. Now, if you remember the Desimone hypothesis, it was twofold. It was focus on the location of attention, but also shrink the receptive field to focus at that location. This one is the shift. We can, of course, also measure the shrinkage by comparing this size versus this, or this size versus this. And there is an effect in our data. It's significant, but it's very small. So there's just enough evidence for a significant effect. But the main effect is that the center of the receptive field is distorted toward the attentional location. But that's exactly what you want. This would give you an enhanced representation to the attended at the cost of losing a response to the unattended stimulus, both inside of the receptive field. So the last thing I want to point out, because we're talking about hierarchies today, right? I wanted to at least have one set of data that link MT more directly to neighboring areas and see what the hierarchy does to these effects. So let's move along the hierarchy. Let's compare attentional modulations between MT and the next area along the pathway called MST. And I have to briefly introduce something to you, which is an optimal stimulus for a typical MST neuron. The optimal stimulus for an MT neuron you already know. It's a linear motion stimulus. We can represent it like this. The optimal stimulus for many MST neurons is called spiral motion, and it's a rotating or expanding or contracting dot pattern. Now, linear motion is easily described by the angle that the motion forms versus parallel lines. It turns out that these spiral motion patterns are equally well described by the angle, or that's just the movement that you already know, by the angle that they form versus radial lines. So you get a similar space, you get a circular space where one stimulus turns into the other. It's a less intuitive space, so I'll show you what these stimuli look like. This would be the continuum of directions. You're starting out, let's say here, clockwise motion, then you're changing that angle and it becomes a contracting clockwise, completely contraction, then contraction counterclockwise, and so on, and the space wraps around. If you've never seen these stimuli before, I'll show you one here on the right. It starts out with pure expansion, then rotation comes in, then ultimately pure rotation, then the contraction sets in, and it becomes a contracting pattern while still rotating, and so it keeps going to almost complete contraction, and then the counterclockwise rotation sets in, and the whole thing just continuously moves through this dimension. The reason I'm showing this is it's nice because it sort of looks like linear motion, but more practically speaking, neurons and MST show the same tuning curves that we see for linear motion in MT to these patterns. And that means we have a nice stimulus to try out attentional modulation in MST. Now, you already know a lot about attention now, so I have to give you the full matrix of possibilities here. So we have spatial attention versus feature-based attention. We have MT with linear motion versus MST with spiral motion. I'll show you the time courses to give you a feel for the attentional modulation, but the real take-home message are the percentages. So this is what you already know. Spatial attention modulates responses to linear motion in MT, and this is the new data. It does the same thing in MST. And the magnitude, and we were really surprised about this, the magnitude is actually almost the same. So we didn't see the expected increase in effects of attention as we move along the hierarchy. It's a small step, so maybe that's part of the reason. For feature-based attention, you already know that in MT you have an effect. This one was a little smaller in that population, but it's highly significant, of 7%. And in the same animals, using the spiral motion stimuli, we find a modulation of about 9%. So no difference here, no difference here. Okay, so the two areas in terms of attentional modulation are the same. Nothing surprising seems to be happening here. No increase of the effect, but also nothing new. And this is until we realized that these MST neurons actually have usually tuned to two things. They're tuned to the spiral motion patterns, but they're also and often equally well tuned to linear motion. Okay, so if they're tuned to linear motion, we can just repeat this experiment in area MST to see whether we see anything surprising there. So I'll summarize those four numbers. You've already seen them. And now let's look in comparison at MST linear motion responses. Again, with spatial attention, nothing surprising, everything is exactly the same. The big surprise was down here. So we really looked, and there is no attentional modulation in MST for linear motion. Now, if you think about this, this is quite surprising because the input to MST comes from MT. In MT, these linear motion patterns are modulated. So we're getting a modulated input into area MST and then it suddenly vanishes, it just disappears? That's odd. We really didn't expect this. Thinking more about it, our current interpretation is that what we see here, or what these neurons represent, is not simply picking up the signal from MT and passing it on. We believe that these neurons represent an intermediate stage of generating these spiral motion patterns. So the computational step. It's not just the signal that they already get. But why wouldn't they be attentionally modulated? Well, if they're only an intermediate step, they don't have any direct influence on perception. They're just used to build spiral motion selectivity. If that hypothesis is right, then we should be able to show that this here is behaviorally relevant and these neurons are not behaviorally relevant. That's hard to do, to show whether single neurons affect behavior or not. One way to look at this is to see whether the strength of attentional modulation affects the reaction time of an animal. So the easy idea is, on trials where neurons are particularly strongly modulated by attention, reaction times tend to be shorter, as if the animal is paying more attention. But of course, this should only work if there is a link between these neurons and behavior. And just sort of as a control, we can look at this for area MT, and we indeed find that selectively or significantly, there is a correlation for the preferred linear direction. And there isn't one, and there shouldn't be one for the anti-preferred direction, because these neurons presumably don't contribute to perception. So that is already a known data set, and we just replicated this. And our question was, how does this relationship look like for MST? Sorry. Here we go. So if we do this in MST for spiral directions, we find exactly the same relationship. A significant correlation for the preferred spiral direction, none for the anti-preferred. If we do this in MST for the linear motion, we don't find a link between the response to linear motion, even for the preferred linear motion, to behavior. So it is an indication, along with the hypothesis I just told you about, that these neurons do not seem to have a direct contribution to our perception of linear motion, while the MT neurons do that. So let me summarize. For what I just showed you for MT-MST, spatial attention does create multiplicative effects for linear motion and spiral motion in MST, and they look the same as an area MT. Feature-based attention works for linear motion in MT and for spiral motion in MST. But it only works for spiral motion in MST. It does not work for linear motion in MST. And there's a good correlation between the reaction times only for the spiral stimuli, not for the linear stimuli. And overall, what I try to show you is that physiologically we find attentional modulation in extrastriate visual cortex. What happens here seem to be changes in gain that create multiplicative modulation, but I also showed you, don't be fooled by the term gain. You can get non-multiplicative effects even with simple gain changes. And I showed you spatial effects, feature-based effects. I didn't have time to talk object-based attention. And I introduced you to the feature similarity gain model as a shorthand for the physiological data that we have collected. I also didn't get to talk about the time course. But overall, sort of on the meta level, what all these effects point is at an enhanced representation of the attended stimulus and a reduced representation of the unattended stimulus. Exactly like what William James was saying, over 100 years ago. Attend to something, it will give you a benefit, but it also means the unattended portion of the input would suffer as a consequence. And we call this an integrated saliency map of the environment, which integrates the sensory power of the stimulus with the attentional modulation that goes on top of that. And I also showed you that this modulation can also affect events like changes in the direction of motion rather than direct physical properties of a stimulus. I showed you that some of it is a shift in receptive fields. We don't know yet how feature-based attention works because you can't do this by shrinking receptive fields. It would have to work in some different and so far unknown way. And the last point I wanted to make is that when you talk to a physiologist about magnitude of physiological changes based on some influences, you have to somehow see how you calibrate or how you scale those numbers with perceptual effects. Thank you very much for your attention. I want to finish with that picture I showed you at the beginning, and I want to point out, to save René Magritte, that of course he realized that this is only one way an artist can be seen. This is the other way, which is much more alike to what I told you about. So an artist, of course, knows that a drawing is a representation, so drawing an egg might sometimes create something very different on the canvas. Thank you very much.
We have time for a few questions. Yeah, Vicky.
So how does the gain change of individual neurons can create this overestimation of the shift in the population?
Oh, you mean for the population response? I think what we're seeing here is an interaction between adaptation and attention. So if you show this one direction movement for an extended period of time, in our case it's a few seconds or a couple of seconds on average, the expectation would be that the neurons who align exactly with that direction get slightly adapted, which will create an overestimation of the change. So that would be an effect where you don't need to talk about attention at all. It's still such that attention increases this overestimation. So the most parsimonious explanation would simply be because the stimulus is attended, it generates a larger response, which generates a larger adaptation, which generates a larger overestimation. Whether there is more to it, we're still trying to figure out. But the consequence of this is still that a change in our environment is going to be overestimated, and it's going to be overestimated if it's an attended change more than if it's an unattended change.
I have a question. So regarding the comparison between feature-based attention and spatial attention, I noticed in your graphs showing the responses that if you look at the time course, if you can go back to that,
sure.
In the spatial attention, you get this modulation right from the start, while in the feature-based attention, it does not kick in right at the start, suggesting that, okay, even if you're looking for something red here, you actually have to see the red to remind yourself, well, that's what you're looking for. That's my interpretation of this.
Yes, that varies a little between data. So you're right. In that data set here, you see the spatial attentional modulation right away.
Right.
We have other data, and you're comparing it to the feature-based. Let me bring that up. Where that is not the case. Here you don't see that. And also in the last one, you don't see that. That's true for this data set, but we don't see this in other data sets. So all of the data I've shown you always refer to this sort of more or less steady state part of the response. And Uri is very right in pointing out this is very interesting to see what's going on here. Unfortunately, the story doesn't seem to be very clear. In many of our data sets for spatial attention, it starts out just like that and only separates later. One of the interpretations we've had is that this is what you expect from neurons who get a very strong on-response, as if they sort of hit some ceiling, and even without attention, it's already hitting that ceiling. So it might be that this population of neuron, on average, had a less dramatic transient at the beginning. So we're looking at this, but so far no consistent story. What you do see here, different from the early data I showed you, is this is smaller than this, and this is something that we've replicated in a way. So in tendency, feature-based attention seems to be weaker than spatial attention. But it also, of course, depends on the task and exactly what the animal is doing.
Yes, Adi.
Maybe to recap on Mickey's variation on this one.
Yeah.
It seems to me a better strategy to actually adapt the non-attended stimulus in order to increase the saliency of what you want to attend to now.
Sorry, what do you mean by adapt?
So there is a gain in what you're attending to, right?
And a loss of gain to the unattended one. It's both. You're right. I mean, in terms of the relation, the ratio between them, making a change on a low number will, of course, give you a much more sizable quantitative effect. But there's been debate from the very beginning, from the very first data that Desimone collected in monkey, whether attention works on the attended or the unattended stimulus. And his early theory was only the unattended is affected, exactly along your lines. From our data, we would suggest that both are affected. You get an enhancement of the attended and you get a suppression of the unattended. in summary, that's the best you can do. If you would only do one, I agree with you. Conceptually, it would get you a little more bang for the buck if you suppress the unattended one. Yeah.
Okay, thank you very much.
Thanks again.
Okay, good afternoon. So that's the title of the talk. And in fact, it will not start with computer vision, but with human vision, and then I will go on to make some connections between human vision and computer vision and some theories of recognition. But let me start with the observation when you do visual recognition. In terms of our own visual system, the human visual system is very efficient in using limited information. We can recognize not only complete objects in front of us, but we can recognize very well reduced configurations in terms of size or in terms of resolution. For example, I guess all of us can recognize any one of these patterns. Some of them are very small parts of the actual object, face or a human being. This is a very small part of it. Some of them are not exactly well delineated parts, like everybody probably can recognize what this is, but it's not exactly a well delineated part. We can all recognize what these small images represent. Now, for the purpose of recognition, the ability to be able to extract information and make recognition, make deduction based on limited information, is very useful in the face of dealing with images which are highly variable. And in fact, when you think about recognition, recognizing things that belong to a similar class, usually the different instances of the same class can be highly variable, and the basic problem of recognition is to deal with this variability and identify what is common to the different instances that come from the same class. Now, looking at the initial images, you see that all of these images have something in common. They all show us people engaged in a similar action. They all engaged in the action of drinking, in this case even drinking from a bottle. But the images themselves, when you look at them, they are highly variable, very different things, very different visual features occur in the different images. If you can focus on smaller sub-images, which are already highly meaningful, they capture a lot of the information that we need in order to identify the action in these images, you can see how the variability is reduced. And in fact, these sub-images are almost copies of each other, and we can focus on those. This will be much easier to deal with in terms of fighting the problem of variability. So if you have a visual system that can already deal with information which is reduced by meaningful, it will be very useful in terms of making inferences and recognition in different scenes and with scenes which have high variability in them. Now, I will talk about some aspects of dealing with this, the ability to recognize images which are minimal for recognition. I will start with some human studies, what humans can do. I will compare what we find in humans with some computational models and end up with some potential implications to fairly broad issues, the kind of representations that we use for recognition, and some of the brain processing that may be needed in order to make efficient use of such minimal images. So these are the people involved in the study. I will not go today into too much detail on this, but let me start and tell you about the human study in which we wanted to collect and to examine images which are minimal for humans in terms of recognition. We wanted to see within large images to find all the components which can be recognized, but are minimal in the sense that you cannot recognize anything more reduced or smaller than those. And we've done the following experiments with human subjects. You saw a subject an image, or a piece of an image, let's say an image like this, and see if they can recognize it. If they can recognize it, you generate from the original image 5 descendants. The descendants can be smaller images or cropped images. What you see here is 4 images. Each one is cropped in one of the corners. So the red frame is what remains from the image, and the image is cropped in one of the 4 corners, and you get a smaller image. Or you can take the original image and make it smaller with less pixels or less points representing the same image. by subsampling it and making a smaller image from the full original. So if this were, say, a 50 by 50 point images, pixel images, you can make 45 or 48, let's say 45 by 45 images, either by cropping it like this, or by keeping the full image, but instead of having it 50 by 50, you make it a 45 by 45. When I say pixel or points, what I mean is sort of visual elements or sampling points, which you can get by, there is a well-defined number of points that you need in order to represent an image, which is given you by the sampling theorem, which is, in terms of Fourier components, it means that you have to have twice the number of points then you have the cutoff frequency in the Fourier domain. So if you have 25 cycles per patch in as the maximum frequency, you need 50 sampling points in order to represent all the points in the image. In principle, you can then make it smaller or larger on the screen. You can make it 100 by 100, but it still contains 50 by 50 sampling points. You can take the image, resample it, take just 50 by 50 points from this larger image, and reconstruct the entire image. So it's a well-defined measure of how many sampling points you have in an image, and you start with a particular image and you make it smaller by cropping or by reducing the sampling points, taking a low-pass filter of it, and if any of these sub-images can be recognized, you repeat the process and you take this image, you generate from it the 5 descendants, all the other ones as well. In this way, you explore a large hierarchy of multiple points at all locations and at decreasing sizes and resolutions in order to find the images which are recognizable but are minimal for human vision. Now, as you can imagine from this requires a large number of subjects, because you can use subject, basically you show them one patch and that's it. Because if somebody can recognize this, you cannot use the same subject and then show them a sub-image. They will immediately recognize they've seen the original and they have it in memory and they can recognize it. Will influence their judgment, of course. So you show them a patch and that's it. You do not show the same subject, the different patches, but you show them to different subjects. So it takes a lot of subjects. So 15,000 subjects participated in this study, and this was done not by bringing them to the laboratory, but doing this online using the Amazon Mechanical Turk, with some laboratory controls for checking for certain things that will be repeatable and controlled in laboratory experiments. And what we define as the minimal images, and that's what we are after in this part of the study, to identify those, find all the minimal recognizable images in an original image, and we define a minimal image as something, a patch, which is recognizable. People tell you what it is, but none of the 5 descendants is recognizable. And let me show you some examples, because one interesting, and it will become important in finding, which was quite surprising and it's quite striking, is that it's a very large, sharp transition between recognizable images and non-recognizable images, almost kind of a phase transition. Things are recognizable, 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 and then you get something which people can recognize, and then a small change in the image can render it non-recognizable. And let me show you, to show you what I mean, let me show you some examples, and to have some fun, let me do it, show you the images to you, and let's see how many people can recognize them. And to do it, I will start with the non-recognizable, the thing which is below the threshold. So this is recognized still by most subjects, and each patch was viewed by 30 subjects. So all the recognition rates that I'm giving you is out of 30. So I'll show you first the sub-image, or the image which is below the recognition. This is, of course, a statistical change, so maybe some people recognize it in the audience. Then I will show you this, and we'll see if more people, significantly more people recognize the more recognizable one, and we can look at them side by side. It's not ideal conditions. Some people are very far from the screen here. Some people are very close to the screen here, and there are limitations, some limitations on the size, not a dramatic one, but there are. So, but let's see how it works. And I will show you, we call them, sometimes when we use this SMIRC, is just an acronym for minimal recognizable configuration, and I call them configuration and not objects, because as you saw, there are subparts and sometimes sort of not very clearly describable sub-configurations of objects. So I'll show you the first one. If you recognize it, just raise your hand. Don't say what it is, because this can influence people around you. So just raise your hand if you recognize it. And so this is supposed to be the the more difficult one. Okay, I don't recognize it. We have one. Let me show you the. Okay, let's see how many people. Not everybody, but a little bit more. Let me show you the two one. This is side by side. Can anyone, more people recognize it? What do you see here? A horse, right. So this is a horse. This is the... Now, out of 30, this is recognized by 93% of the people. This is by 3% of the people. And you can see that the differences are not large. There is a small difference between the two images, and it makes this difference in recognition. Let me show you. Okay, this is the full part of the hierarchy. I told you about the 5 descendants that you make from each image, recognizable image. This was still recognizable. These are the 5 descendants, and you can see that the recognition drop. It can drop by small change to 3%, 0%, and so on. Let me show another example. Anyone can recognize this? Nobody? Okay, let's see if yeah, what do you see, Daphne? What is it? Oh, anyone else? What do you see here?
A ship.
A ship, right. Let me show you the ones one by one. So this is from 80% to 79%. So this is recognized by 80%, this is by 0%, so here too 0%. I don't think we went all the way up to 80%, but more than half of the people recognize this. So you can see again that the differences are not large. One last one, and then there are hundreds and hundreds of those. It's the repeating situation. In all of them, you have this sharp transition, and I'll just show you one more for various reasons. Anyone here? Okay, at least one, maybe two. Yeah, let's see if this is.
Oh yeah, easy.
Now this easy. And look again at the differences. These are the two. It's 90% and 15%, and you can see that the difference is this here, right? Which is in terms of pixel and so on. It's not very large, but it's very meaningful. We need it. It makes a big difference, and here. It worked very well here too, that one person recognized this, and virtually everybody, maybe almost everybody recognized this. So we started this entire experiment with 10 different images. These are the images, and we wanted to recognize all the minimally recognizable configuration within each one of these images, and we did it in the way that I showed you. And here are some examples of, we ended up with a full database of literally exploring this entire hierarchy, what people can recognize in these images. You see in each one of them the percent recognition, and also the size of the image, which I'll mention in a minute. I'm just collecting here. We saw 3, just to show you a few more and to look at them. It's again, it's striking and how small the differences are in what they are, which is the next point to look at. But for example, you can. This was a part of the horse, the same horse, and you can see that it's very important if the top of if you see the ending of the hoofs of the leg. If you see the ending of it, if you cut it and you cannot see it, and that's very different. And in all of them, in some of them, it's almost difficult to say what exactly is missing, but the difference is. Difference in recognition is large. The difference in terms of the actual features you extract and use for this recognition is small. I found it interesting. This was not the main point of the study. I found it surprising the sharp transition shows something about the uniformity of our visual representation in our brains across different people, because here we do not, the differences are not just within one person, that the person maybe does not recognize this, and then you increase the image a little bit and suddenly they see it, and the individual has a sharp transition. But that's across everybody, meaning that if you just show it to people, and these are people online, different countries, different states, different ages, coming from different background. And basically almost everybody, more than 90% of the people recognize this, and virtually nobody recognizes this. It means that everybody has the same representation in their head of something very similar that allows them to extract from this that this is probably coming from a horse, but they do not see it here. So there is a very striking uniformity in the kind of representation that people develop in their minds.
But it must be age-dependent, culture-dependent, right?
Sorry?
It must depend on age and culture.
It must, but you see that it's 90% to 3%, right? So it's sorry? No, these are not students because it's online. These are people from India, different people from the United States. These are so I think that they're also horses in some sense, but probably there are features here which are important that the visual system extracts and are useful, and it's much more uniform than you might have expected. If your individual experience made a big difference, then things will be more smeared, that some people will see it here, some people will see it, we need a larger image, or we'll deal with a smaller image, but it does not seem to be the case. That's the reason I sort of said almost phase transition. It's sort of everybody collapses to a different state. Yeah.
What was their choices? Did they have a list of items?
No,
that they could
it was open recognition. They see a patch like this, and they have to type what they see.
So if they would write animal,
okay, so right. So I can tell you there are some animal would not accept it. So I'll tell you the way we did it, and we designed it from. We had rules, and we followed these rules, which were very, I think, they are simple, and I think they make sense. We used, in order to decide. Suppose there is a fly, there was a picture of a fly, what do you do if somebody says a bee? Is that okay, or this is not okay? What we've done is that we took, there is a hierarchy of nouns in the English dictionary, which is called WordNet, and it's organized hierarchically. So you can see if two names in the dictionary are considered siblings or are more farly removed. And we accepted siblings, and it turns out that bee was accepted for a fly, but a spider would not be accepted. So there were rules, and there are very, here and there are some things that one could argue about, but there are very few. And typically, when people did not recognize things, typically they wrote none. We allowed them to sometimes they guessed, and it was considered wrong. So you would say, I don't know, a flower or whatever, but it happened, considered wrong. As I said, if they gave a noun, we checked, and the noun was what we accepted, a horse, that was okay. If not, we checked it if it was a sibling in the dictionary. We said yes, and that's the and that was the scoring system. In terms of sort of visual elements, how many pixels you have in a recognizable image, the numbers are small, are typically less than 25. I'll show you some of them. We had in one or two, they had a bit more than that, never more than 30. So the images are very small in terms of the number of visual elements you need in order to recognize them. You cover, or these minimal images cover the original image in this way. You can see here that the original image is tiled by multiple minimally recognizable configuration in terms of size and positions. Some of them are large and cover almost the entire object. Some of them cover only a small distinctive part of the object. And again, don't confuse here size with sort of the number of elements. It turns out that it's sort of universal, that you need about 25, roughly speaking, upper limit of 25, maybe 30 points. It doesn't matter if the entire object, you can have a low resolution copy of the entire object, which is say roughly 30 by 30, or you can have a higher resolution image focusing on a smaller distinctive part of the object. It can be a 30 by 30 image of just the head of the eagle or a corner of the eye and so on. There are quite a large number of individually recognizable different images, sub-images, which if you have a system that can effectively recognize these minimal images, and you'll see in a minute that it's not that easy to get an effective system like this, then you get a lot of redundancy, because you have here, say, we have, on average, we have almost 17 of these individually recognizable images within each one of the original images, and each one of them is recognizable, and it tells you what it is, or tell you roughly what it is, or almost exactly what it is. So you have a lot of redundancy. Things can distort, things can be cluttered, things can be occluded. as long as you have some of them still visible and recognizable, you will be able to complete the recognition task. Now, the striking thing is this sharp transition, which tells you that there are features inside the image that we are very sensitive to, and you get an opportunity to try to look at them and find what they are by looking at the recognizable, what we call the minimal image, and the sub-image, the image that you can recognize, and then another image which is closely similar to it, almost identical, something is different, but it becomes non-recognizable, and by analyzing what you missed in the sub-image, which made it unrecognizable, you can start to get information about the kind of features which are critical to the recognition process. So in this sense, it sort of provides one with a sensitive tool to compare representations, to see which representations can capture the difference between the recognizable and the unrecognizable part. And as people deal with computational modeling and so on, we were intrigued to see whether existing models can capture the differences, can recognize these minimal images, and will they be sensitive to the kind of differences that we humans are in the transition between the recognizable and non-recognizable images. So this is not a story in computer vision. If this was a computer vision talk, I would go into the details, which I think are interesting, of what are the leading models, what are the strengths and differences, weaknesses of different ones, what exactly we can test here, but I will give you the gist of it. What we wanted to see, we took a number of models that I will mention in a minute, and we trained them, and then we tried to test them to see if they, after trained on pictures of a fly, in this case, what will respond to images like this. Would they be able to recognize minimal images at all? Would they show the sharp transition that humans exhibit between, at a certain point, between the recognizable ones or not? So we are interested in the recognition gap between successive images like this and the accuracy. Can they at all recognize reliably minimal images in the sense that we do it? And we did a number of testing. The first one is a difficult but natural one in the following sense. That what we train the classifiers, the models on, are complete objects. We do not show them small patches of images to sort of focus their attention on small patches. But for example, if airplane was recognized by something like this, which captures a part of the tail, we train the recognition classifiers on complete side view of airplanes in this way, and there are some wonderful models around that have good performance. I will tell them about the best ones in a minute. You test them, you train them, you test them that indeed they recognize airplanes very well, and then you test their capacity to recognize these recognizable subconfigurations. And this is relatively natural in the sense that we are not trained explicitly, we are not exposed specifically to small subconfiguration. We typically see the object, or at least a large part of the object, and we still develop internally, we extract the relevant information, it's sufficient to make us capable of recognizing meaningful but reduced configurations. I will mention the names of the models. It will not say too much to those of you who are not in the field, but I'll tell you in a word or two what they mean. These were until 2 years ago sort of the leading two models in the world in the ability to recognize, to train on an object and do visual recognition. One was called a deformable part model, and the other one is something which is called variation of bag of words, two great models. The last one, let me mention first, it's called HMAX. It's sort of the leading model around for trying to model directly the ventral part of the human visual system. So this was developed at MIT by Tommy Poggio and his colleagues, and they combined computational algorithms with data, which is all the data we know about V1, V2, V4, and the various stages in temporal cortex, and they build a model that on the one hand It's very consistent with the anatomy and physiology, and on the other hand, it's competitive with the best models that came out of strict computer vision. So it was interesting to test this human-inspired or biological modeling. And the other one which is mentioned here is deep convolutional neural networks. I'm just wondering if how many people here heard about deep convolutional neural networks? Okay, about half. I can tell you, at MIT there was it made such a big buzz in the general literature, certainly in modeling and brain and vision, but at MIT there was a seminar on recognition and so on, that typically 30 people come to the seminar. There was a seminar that talked about deep convolutional neural networks in recognition. 650 people showed up. And it's typical. It's something that changed in the last couple of years, the way people do modeling in vision, in computer vision, but people also suggest it as potentially something quite interesting and perhaps relevant to biological modeling. It's a new type of way of training neural networks for the purpose of recognition. They reached better performance than the previous models, and are still being developed. So in the States, there were papers in the general press, in the New York Times, and everywhere, and they're really doing something interesting. Perhaps less than what the open literature ascribes them, but they are doing interesting things. It also made a very large commercial impact. The first paper that came out with and showed improved results in recognition using this deep network was by Jeff Hinton. People here who come related to cognitive science may recognize the name of Jeff Hinton from the old days of connectionism and backpropagation and things like that he was one of the developers of. So the paper came out a week after that. Google bought him from the University of Toronto. He left the University of Toronto. He's now a Google person for many, many millions. The week after that, there is the rival of Jeff Hinton is Jan LeCun from NYU, who is also dealing with, in fact, he was in a sense the first to develop these networks. And so the week after Google made the move and bought Hinton from Toronto, Facebook made a move and bought Jan LeCun from NYU. Each one of them each one has large laboratory of hundreds of workers that are doing this kind of work for the industry. So that's the story of this, and we included some of these sort of modern deep convolutional networks in the analysis as well. This is some visual rendition of HMAX, of the biologically inspired one, and the other one is the deep network. So I will not go into the detail. I'll tell you immediately the bottom line, and I'll show you just some graphs supporting this. But none of the models, including the deep convolutional neural network and HMAX, none of them replicated the gap. They do not show anything like the gap in recognition between recognizable and non-recognizable one, and none of them produces good results on simply the task of recognizing these minimal images. They're unable to do this compared to human vision. To show you, this is which one? It's one of the previous model, DPM. What I'm showing here, just to show you what I mean by the results, this bar on the left is the difference in recognition rate between a whole collection of this is all the recognizable images of the horse, and this is the sub-images. So this is just the results of the human study. After training on horse images, you test this image, and the difference between the recognizable one and the non-recognizable is this, which is barely reaches significance. It's very small, slightly less recognition on the images that people could not recognize, but the gap is not reproduced. This is with the deep recognition network, which is sort of now it's the leading kind of model. You can see again, just showing here again the human gap between recognizable and not recognizable. In the deep net, it's actually going in the opposite direction. Now, one has to make a distinction here between this, the gap, and overall or absolute performance. This does not indicate directly what is the performance of the model, because what happens here is that we set up you have a model, and the model gives you some score on how much it believes, or how much of these are bicycles, how strongly this belongs to the class of bicycles. Now you can set an acceptance threshold such that, say, 50% or 70% of the images will be above the threshold. You can always play with the threshold to reach a certain recognition acceptance. The question is how many non-class images you will accept, how many mistakes you will make. It's the trade-off between correct recognition and false alarms. You can always get more and more correct recognitions, but at the price of having more and more false alarms. So what we see here is that the acceptance rate was artificially set at such a rate that the model and the humans will be the same, that both recognize and accept as correct bicycles around 70 or 70-some% of they're almost matched for technical reasons. It's not exact, but it's close. And then at this level, you can see that the same number, or even slightly more, of the non-recognizable bicycles are accepted. But if you look at the accuracy of recognition, this we see on this graph here, the one I'm showing here. Blue shows the correct recognition of full images, namely, after being trained on bicycles, you show the model new bicycles that have not seen before, and you can see that the recognition rate is close to 100%. They did very well on recognizing new bicycles, but the recognition was about 3 or 4% when you show them or the average precision. When you compare it with non-class images, then the accuracy in recognition dropped from close to 100% to about 4%. So the accuracy in terms of recognizing minimal images is very, very low, and the gap is not reproduced. So this is the summary of this. The recognition of minimal images does not emerge by training any one of the existing models. It seemed that the humans, these tiny differences that make the difference between the recognizable and non-recognizable images, show that the human visual system takes some local and informative and useful features. It gets, using them, high recognition results. It pays the price, so maybe it's not a price, but it gives rise to this sharp difference when these important features, discriminative features disappear. You can no longer do the task. The interpretation here is the models do not extract, do not use these features. They have a different or not identical representation to what we do. As the result, the two properties emerge, that they do not show this gap and they do not make a good recognition of the minimal images.
Is there a threshold with, let's say, deep neural networks? With other features, I mean, maybe the threshold is different. There is a threshold.
I mean, you put the threshold to get 70% to get the same kind of recognition.
Then you reduce the resolution one way or another. Maybe
you do not reduce the resolution. You take these images and you see what kind of these patches give you a certain score. You make sure that 70% of those are above the score. It turns out when you do this, then if you take random patches in the world which have nothing to do with bicycles, a large proportion of them will also be above this recognition rate. And the fact that you cannot get the gap is that you look at the scores that the classifier, the models give to these images. On these images, they are entirely intermixed. If you want to recognize some of those, you will recognize invariably also some of these. So both of them, the scores are not very meaningful. They are not much higher than some relatively random patch, and are entirely intermixed. So it shows the limitation of this. It's not the kind of representation that we produce, which allows us to distinguish between these and non-bicycle images, and it also introduces big differences between these two levels. So this is what happens in the model. Now we gave them the models the benefit of the doubt, or we gave them some tailwind, if you want. You can say that, well, it can come from two related deficiencies. One is that maybe the model unlike us, somehow you give it the large image, it doesn't know how to focus its attention on some relevant parts and try to extract the meaningful features from these important regions. So it didn't generalize from being trained on the entire larger airplane image to the variety of sublocal images which are informative. It couldn't use it. But what will happen if you train it specifically? If you want to recognize something like this, you do not wait for it to emerge from the full images, but you cut a sufficient number of training images which all focus on the tails of airplanes, and you give them, and you now train, you go through the same exercise of training and testing, but not generalizing for complete objects to meaningful subparts, but training specifically on each individual part. And this requires another pretty large human study, because when we give the models specific subparts, we want to make sure that when we cut the equivalent, say, tails of the airplanes or parts of a fly from different images, that all recognizable, but we want to compare human recognition with the model recognition. So we also went one by one with all the additional patches that we cut, and we tested human recognition and all the additional training images. You can see one example of the many minimal images that are involved. So this was from the original image, that this part of a fly was recognized and this part of the fly was almost non-recognized. And these are many, many other images, and you can see the recognized one and the unrecognized one and so on. So it requires also some additional human testing. And although the results improved in terms of correct recognition, we see a significant boost in recognition. If it was on average in the 7 or 8% correct recognition in training on full images, it was between 65 and approaching 70, which is still way beyond humans, but at least when you recognize them, when you train them explicitly on sub-images, they produce better results. The gap, this is from one classifier, the gap didn't change. The gap between the more recognizable and less recognizable remained the same. And finally, okay, there was a number of other tests. One other test that I will mention was testing and training humans on exactly the same images that the models are tested on. In doing this, unlike the open recognition test that I mentioned before, here what we do is we show 60 images to humans. Some of them, these are not the best images I could have shown, but these are taken from the torso of a horse. Humans get these images. Some of them were actually from horse images, and some of them were similar images which are non-horse images. And humans just look at them and they're asked, mark which one of these images are images that come from a horse and which ones, in your view, do not come from a horse. And they have to mark each one of them as horse, non-horse. We do this for 30 images for each one of these patches. There are many patches to test, and the same thing is tested using models. And again, the main result is that the ability to recognize minimal images by humans is much, much better than the ability to recognize. So you can see the different classes and different images. So zero errors for humans, 17% errors for the model, and so on. And 17%, by the way, you have to appreciate that making 17% error is terrible. It means that it's useless, because what you show is you show a number of patches, say 1,000 patches, and on 17% of them, the model says that these contain a horse patch. Now, if you think about the number of small patches that you have in your visual field, and what happens when you look at the world around you for 3 minutes, then this kind of performance means that the system will be swamped with thousands of false alarms from each class that you know about all the time. So it's really unacceptable. Now, let me sort of shift gear, tell you some related thoughts that or the direction that we are now following. in terms of computational modeling, in terms of some brain testing, and I don't know how much time I have. But, 10 minutes. So I'll do it briefly, but to let you, to tell you a little bit about this direction. We think that what we see is also related to something else that humans can do very well, and computational modeling now cannot do. They still didn't get to this stage. They simply cannot do it. And this is to not just label an image, whether it contains a horse, yes or no, or an airplane, but to give us what we can get, which is a rich, full internal interpretation of what we see in the image. And it's interesting that we can do it for these minimal images as well, because if you look at an image like this, or an image like this is I don't know if you can see it, this is maybe not the greatest one, that is a man in a suit and tie. When we can recognize that this is a man in suit and tie, it's not just a label, but we can get a rich set of internal details inside this image that we recognize. Can everybody see that this is a person? Okay, and we know that this is the neck, and this is the tie, and this is the knot in the tie, and this is his shoulder, and this is a part of the jacket. And in fact, we tested it. We asked people just, annotate this image. Tell us what you see, mark what you see in this image. Again, online, and this is the kind of thing that people produce, following our instructions. So people can get inside this minimal image, and it's sort of, I call them atoms of recognition, but it's not atomic in this sense. So it's atomic in the sense that you cannot make it smaller and still recognize it, but you can still go inside and recognize a rich structure of things that you can recognize inside. And this is part of vision. I mean, we are not going around just labeling gross things that there's an airplane here and a horse there, but we can see a rich set of details almost everywhere. We know almost every pixel in the image what it belongs to. Now, once you can do this, or interested in, and you can do this internal interpretation of what goes on inside the image of interest, then this sort of automatically gives you also a possibility of improving the recognition accuracy and doing a better job, because if things which are perceived as a person in a suit by the leading model that I described to you, you can reject them if they lack the internal structure that you are looking at. If you're looking at and if you think that you discovered a man in a suit and tie, then this kind of internal structure should be apparent in the image. If it does not, it's not a correct recognition. So we think that this is an important and unexplored and interesting challenge, the ability to go beyond labeling images as the kind of type of class of object they contain, but to be able to perform the complete internal interpretation, and that's an important goal on its own. And as a side benefit, a likely outcome would be an increase in performance. When we look at the kind of internal structure that distinguishes correct images from incorrect images, the kind of features that are required in order to make this distinction seem to be more challenging and more complex than the kind of features that people are now using in all the computational models, including the deep network one. They seem to be required to do certain groupings of small elements into a long contour and seeing what is the curvature of a contour, or they need to do some grouping in order to see what is the texture inside a region of interest. So what is the repeating texture inside the horse image compared to non-horse images. This is based on computational experiments. In our hands, the ability to be able to recognize things as horses here and reject these as non-horse depended on things that required us to look at extended contours and to do some local grouping of the internal page and so on. The way this leads to, I would think, is that the recognition interpretation process is likely to be divided into two stages. I think that what people are producing now as these deep convolution networks and other models are in fact probably, they may be close to the truth in terms of describing what goes on as sort of the initial bottom-up recognition stage that happens in our hierarchical vision system as well. In the next stage, you have to apply these grouping and segmentation, paying attention to more complicated features, which are unreasonable for computational reasons, to include in the as a part of the feed-forward recognition process. So the way that we currently think about it is the way that recognition and full interpretation of the image goes is roughly like this: that you have the image, you pass it through an initial bottom-up phase, which produces good candidates for initial recognition, but they are not the end of the story, because they still would make, as all the models I've shown you still make, they will still make a large number of false detections combined with inability to perform the internal interpretation. The way that it will work from that stage on is once you have a particular class being triggered by or being activated by the bottom-up stage, it will trigger something which is more top-down in nature, which will now it will now concentrate on specific images and specific location with these images, so it can apply some more sophisticated feature detection and testing that you cannot allow yourself to build into the bottom-up stage. So this will, the activation or the initial activation by the bottom-up process of a class candidate will trigger the application of extracting additional, more complicated feature and complete the recognition process together with performing the internal interpretation. And this can expand outward. It's not just filling in the details, the internal details of what you already discovered, but you can expand now, once you have identified correctly the internal features of what you successfully did, you can expand outwards. For example, I don't know if this will be recognizable to you. Can people see what this shows? אלו, מה רואים פה?
רואים אישה לוקחת בקש מפוסט.
אז זאת יותר, זה נכון שיש, אוקיי, it's close enough. אם תראה יותר מקרוב, it's actually there is it's a glass, it's a transparent glass rather than a cup. But you can see the relation to the mouse is very important, for example. If it were not here, but somewhere here, it will not be a person drinking. So, I'm going to show you just an example. Or, I will not. I'll finish this point and go to the last point and then I stop. We think that in order to recognize complicated things like what people are doing, how people are interacting and so on, you have to pay attention to the small details. The small details can be very ambiguous on their own, but that's the kind of thing that we are working on to start the recognition of the level of this independently recognizable atoms of recognition. You recognize them bottom up. This triggers the application of top-down processes which complete the recognition and produce the detailed internal interpretation. This propagates outwards to also interpret and recognize details which are not recognizable on their own, but become recognizable once they have the context of these minimal images which are recognized on their own without context. These are some initial demonstration of the modeling work, which I will not go into. And just for the last 2 minutes, just tell you that we are also starting to investigate this kind of the question, specifically the question about the bottom-up and the top-down components of recognizing minimal images. We think that testing top-down effective recognition, for testing top-down effective recognition, these minimal images provide an advantage, because if you have a full image, it's so redundant and you have so much information that even without the top-down process, you can have a quick idea that you are looking at a horse, even if many of the things are still not entirely recognizable and incorrect if you recognize just a part of the minimal images inside it. So this is a study that is being conducted at MIT in collaboration with with two people at MIT, doing the recognition of minimal images in a MEG, where you can try to follow the, you can try in a high resolution in the temporal domain, you can follow the detection process. And what this shows is what we are doing is we are training a classifier to try to look at take as input the electrodes from the MEG and produce a classification, in this case of an eagle. And we have an initial results which look interesting and promising, because they're very different from what you get in a MEG if you do not go to the level of the minimal images, but you have complete images. You do start to get the correct identification of what the class really is at times which look suspiciously long for a standard recognition process. You get something here that the real classification of this as belonging to the class of eagles is something which is about 300 milliseconds. Another example for the bicycles, again 300 and something. It's very different from the pattern you get when you look at complete images. So we hope that this is only a very beginning, but just to show you that it's also interesting. One can go into some brain imaging and to try to get into the details of recognizing these initial small images and the subsequent stages, maybe distinguish between bottom-up and top-down processing and get the more complete pictures. Okay, so I think that the recognition model so far focus primarily on this first stage, which is important, but it's really only a part of the full story of visual recognition. The model itself suggests that we start the recognition process by independently recognizable images, which are these minimal images, followed by an interpretation and validation second stage, which is more driven by sort of class-specific top-down processes. We are continuing to analyze the images, the recognizable and sub-recognizable images, as a part of trying to better understand more exactly what are the features that the visual system uses for recognition. It's interesting to consider the neural circuits involved, especially in the top-down processing, and we are starting to look into this, and to try to understand not just the individual recognition in each one of these minimal images, but how they can serve as a seed which propagates to the full interpretation of the entire image, and apply it to things which currently are beyond the capabilities of what can be done computationally. And these are complicated images which include action recognitions, interactions between agents, and complex images like this. Okay, so thank you for your attention.
So we have time for two.
You have the weird phenomena also in this computer, where you look for computational algorithm, maybe patch that you can, the computational algorithm would recognize in 90%, but the moment you start doing this cropping or under sampling, it go down to 3%.
We tried it, as I said, with doing it as a connected with the recognition of real images. I guess it's the only way that makes sense. I mean, I can take just a patch and then take a reduced version of the same patch, set a threshold so that for this particular patch, one will be above and one will be below, something like this. But what happens here is that it's exactly what we tested, in which you let the...
what I have in mind,
oh, I'm missing the question, sorry.
You train, say, on big images, and then you do the same psychophysical experiment you did on the classifier. Would it produce the same results?
I think that's what we did. Maybe we should take it offline. But we trained it on the big image. We trained it in two ways. One was on the big images. You take one of these networks. We train it on, say, 500 airplanes and many non-airplanes images. You test it on new airplanes, and it does wonderfully. It recognizes all of them. You test it on subparts which humans can easily recognize, and it falls apart.
Maybe because it owns some.
Sorry? Okay, you mean you can train, you can test it on many different patches and see if you get, we tried some of this, yeah, you never get it, I mean for it, and the reason is, when you look at the representation that emerges, and the representation of a patch and the slightly subcropped patches are too similar for it. You run it through the classifiers and you get the, you look at the scores of the patches and the slightly reduced patches, and they are too intermixed. I can show you some of this results. But I think that we really tried it from different ways of training and testing and so on. And it's the kind of features that you have to extract in order to do the interpretation and to get these differences. All these models are simply not sensitive to, and you cannot get it out of memory. You will just not produce it. Yeah.
Can you obtain primitives that are small, like small group of neurons, for instance, like some primitives that sensitive for directionality, like we saw from the previous lecture?
If you can get such neurons in the model, yes. But the question is, yeah, you can get such.
No, primitive, like some features, like using like computation tools that actually correspond to very small, low layer primitive that correspond to a small group of neurons.
Yeah, no, mate. I can tell you that the best we can, at least the number of models that I mentioned here in the deep networks that I mentioned, it's not that easy to analyze which neuron correspond to which feature. It's easy to analyze or to test the overall performance of the network. It's not that easy to stick your electronic or your software electrode inside the neuron, inside the model, and know exactly what it responds to. The extent that we can, we've done it to some degree, and it's not easy.
Yes, but you can make a list, and then you can actually make an order in the list, and then you can actually hypothesize which kind of primitive you expect to find in the brain. So it can be a tool for prediction.
You can try, and we've tried out of this, some of this, and as I said, at least some of the features that we see as crucial to make a correct recognition and discriminate between the correct patch and a patch that comes from a non-class image requires us to do some grouping and some some segmentation. For example, it's not enough to test for certain texture in the image because it can come from the background. You want to say that within a certain region I expect a certain texture, and for this you have to delineate and make some distinction between what belongs to the figure and what belongs to the ground. We found that you have to do things like that in order to get sufficiently powerful discrimination between correct and incorrect patches. We think that the brain does it, but it probably does it not on all the possible sub-patches everywhere in the image, but it does it once you have a candidate result, and you say, I think, based on the initial recognition phase, I think that this is the head of a horse, then this triggers certain processes which are not done automatically over the entire image, everywhere in the image. So I think it makes sense computationally. If I'm computationally, I think that's probably, and it's related to what Tali, for example, you guess, and then you have to make some checks on your guess, and the checks can be more complicated and not something that you can incorporate freely inside the initial guessing stage. It will be unwieldy. You have to do too many things. So the strategy of guessing, proposing candidates, and then applying something which is specific to this hypothesis, to this candidate, saves you a lot computationally. It seems that that's the reason why it may be divided into separate cycles of producing the candidates initially and doing in the second stage things that you wouldn't do wouldn't incorporate in the bottom-up stage.
I wanted to explore one remark you made when you showed the picture with the guy with the tie and the jacket, and you were pointing out that even on these very small patches, we have this sub-image, parcellation into components, and it's very natural, and it worked perfectly. I would expect that the models don't do that, because the models recognize a plane, and they either recognize a plane or they don't recognize anything. They don't know about wings or wheels or anything. So it seems intuitively that we have objects on all these scales, objects that are parts of larger objects like the wings and the wheels and the engines and so on. And that might contribute to the difference between the models and our perception, because we know also about the relationship between these objects by themselves, which the models, because they always see full planes to learn with, they never have just wheels or recognize that a wheel on a bicycle and a wheel on the plane, functionally are related. They can't exploit that. So do you think that?
I think it's part of the story that you have to recognize the details on all of these levels in order to make, and it's not only to be more correct. You are interested. You look at the dog, you want to see what is wagging its tail, whether the teeth are now showing up, and you're interested. You go to a car, it's not just a car, you want to find the door handle and all that. Yeah. So interacting with objects and all of this. So it's part of vision in general, I think, being able to recognize all of these details. It's interesting that it's not enough to recognize, it's not enough to train, for example, models on each one of these parts individually. Say, well, I can recognize an airplane, now let me train this model on airplane wing or something. Because as you saw here, the thing you recognized was indeed atomic. You cannot train it on the tie alone within this, because the tie alone is unrecognizable. It will be confused with many, many features that can arise in non. So there is something which, a collection of things that you have to, minimal things that you can, it's not that you can train on each recognizable thing on its own. There are some configurations which make a sufficiently large unit in order to recognize it, like the man with a little bit of the neck showing, and the jacket, and the tie. This is enough to recognize what this configuration is, and you have to take this as atomic and then dive into it, and once this is recognized, dive into it and make sense of the components.
But I wanted to make a suggestion. It goes in a different direction. I understand that it's difficult with the models, but how about we look at a human ability that lacks the scaled versions of objects. Mm-hmm. Now for that you would have to train humans on abstract objects, because natural objects all have that scaling. But for example, if you would train somebody on something similar, let's say, to Chinese characters, mm-hmm. so they don't have any natural grouping that we experience, because we only see them in the training. Would you then still expect that gap? Would you have the gap in the human perception go away if the object only exists on one scale?
It's an interesting question. I think that it's a complicated story, so I do not have a simple answer for you. But I think that this is a rich domain we'll have to explore, the interaction between the components and the recognizable things. How do you make it? How do you extract it? Why they do not come up with why the sophisticated networks being trained on images do not, even when you train them on patches, they do not have the capacity to sort of sift out some of these important features. So I think it's an interesting domain that we are starting to look at. Yeah.
These were all subsets of the picture in the same quality as the picture. Is that correct?
The same quality?
In other words, it wasn't a lowering of contrast. It wasn't an adding of noise.
No, it was reduced resolution compared to. So one thing that changed is that the same part could be, you can take the entire image and reduce the resolution of showing it and ask, is the reduced resolution still recognizable? So resolution, sort of blurring, did occur here. Other than that, no additional noise or reduced contrast, you're right, which would affect the recognition as well.
Okay, thank you very much, Shimon.
Okay, thank you.
I'd like to thank all the speakers today, and especially thank Cheryl here, who actually helped us conduct this day. Thank you very much.